Hey everyone, welcome to this video where I describe your DNA replication discussion. For this, we're going to do a DNA replication project, and if you want to watch this video, obviously that means you clicked here. So your task is to demonstrate your knowledge of the process of DNA replication by describing the steps in order and describing the enzymes that participate in this process. Background knowledge. The genetic code is a sequence of DNA nucleotides in the nucleus of eukaryotic cells. Before a cell divides, the instructions are duplicated so that each of the two new cells gets all the necessary information for carrying on life functions. Cells pass on their genetic code by replicating their DNA. During DNA replication, enzymes unwind and unzip the double helix, and each strand serves as a template for building a new DNA molecule. Free nucleotides bond to the template, A with T and G with C, forming a complementary strand. The final product of replication is two identical DNA molecules. For this project, you will choose the format that you want to choose. You have a choice. You could do a journal entry, an instruction manual, a letter, a poster, a mini poster, a drawing, a video, a Google Slides slideshow, a short play that you record, a 3D model, a newspaper article, a web page, or any creative option approved by me. But you have two main requirements. You must correctly describe the process of DNA replication. You must also include descriptions of the following enzymes: helicase, primase, polymerase, and ligase. For resources, you can use any resource that you found or seen in this module or online resources approved by me. And here is a little rubric for your project. You have to have all four enzymes described completely. If you don't, you'll lose points. And you should completely describe the process of DNA replication by using the proper vocabulary and using it correctly. There's a list of vocabulary words there that you might use: replication fork, leading strand, lagging strand, 3' end, 5' end, enzymes, nucleotide bases, A, T, G, C, RNA primer, Okazaki fragments, and semi-conservative. And you would receive full credit if you use all those vocabulary words correctly. If you use less than all of those vocabulary words correctly, you will lose points on the project. I'm including here a couple sample projects. So let's say that you're into writing. If you're into writing, you might make a letter about DNA replication, like maybe Dear New DNA. I'm so excited for you that you will get to experience replication soon. It seems like a scary process, but you will be okay. Actually, you will become even better than you are right now. In fact, you will end up being part of two new cells instead of just being part of one cell. I want to describe the process to you so that you won't be scared when it begins. First, you will be separated into two different strands. Your double helix will be unzipped by the enzyme helicase, which will break up your A's and T's and your G's and C's. This means that your double strand will be separated. Each strand will go on to make up half of a new DNA molecule. All of this is happening at a place that scientists call the replication fork. At the replication fork, of course, there are your two strands of DNA. The one that is going toward the replication fork is oriented 3' to 5', and it's called your leading strand. The other strand is called your lagging strand. Something a little different happens at each strand. At your leading strand, the enzyme called primase makes an RNA primer to attach to you. Then DNA polymerase moves along you, attaching a new A to every T on your strand, a new T to every A, G to C, and C to G. These are complementary base pairs, and they are added continuously. At your lagging strand, things happen a little differently because DNA base pairs can only be added in the 5' to 3' direction. Many primers are made instead of just one. Then DNA polymerase adds complementary bases, just like it does on the leading strand, but it's only adding small chunks of bases at a time. These chunks are called Okazaki fragments. Since the chunks will be need to be connected later, this is considered to be discontinuous. An enzyme called ligase will connect the fragments into one long piece of DNA. Eventually, all RNA from the primers will be replaced by DNA nucleotides. Ligase makes sure that both the new strands are sealed up and ready to go. but not before the new DNA is proofread for accuracy. Finally, the two new DNA strands, which are both halfway made of you because you are the original piece of DNA, this is called semi-conservative, they twist up into a helix shape. It's really important that everything goes right so that your cells will not end up with mutations. Best of luck. Good luck. Best wishes from old DNA. Or, hey, let's say you're not that into writing. Maybe you're not. Maybe you're more visual. You could provide some sort of a diagram like the one that I provided in your notes. Now, you cannot take a screenshot of my notes and put that as your own project, but you could essentially draw this on your own, make your own version, and make sure that it shows all of those key vocabulary words, and also not just shows the words but describes what they mean. So, like, for example, I didn't just put helicase here. I put this is an enzyme that separates the two strands of DNA. So make sure that if you're doing it as a drawing, that you're also just making sure to describe what you need to have there. And when you're done, you can submit your discussion board, which is this project. So you do not need to do multiple projects. I just have a couple here as different samples. Like, if you're into writing, do something like this letter. If you're more visual, do something like this picture. But you get to choose your format. And if you're having trouble uploading it to Canvas, you can always email it to me directly. Thanks for listening.